Hello everyone, my name is Jiayi Xu. I'm glad to present our work entitled Geometry-Driven Detection, Tracking, and Visual Analysis of Viscous and Gravitational Fingers. My co-authors are Shumo Data, Wenbin He, Joachim Morgate, and Han Wei Shen. We are from the Ohio State University. In this work, we study viscous and gravitational fingering phenomena. For example, when pouring honey into water or making a lava lamp, the viscosity or density contrast between fluids causes viscous and gravitational flow instabilities and results in finger-like features. The time-varying data in this work are generated from a simulation of carbon dioxide storage and is applied for global warming mitigation in earth science. In the simulation, CO2 is injected from the top of a rectilinear aquifer. CO2 volume tends to spread out from high-density regions to low-density regions. Hence, high-density regions usually form the center of the finger structures. The simulation finally outputs the time-varying volumetric data for CO2 density over the entire aquifer domain. We interviewed earth scientists for their analysis requirements. They were interested in spatio-temporal exploration of fingers. For the spatial exploration, they wanted to identify fingers and branches in space automatically. Also, it was important to extract the connections between branches. The connectivity of branches might help them develop new scaling laws for the analysis of flow instabilities. For the temporal exploration, scientists were interested in the formation and evolution of fingers and branches. They wanted to identify the growth, merging, and splitting events of the time-varying fingers. In the following, we present the details of geometry-driven solutions for the spatial-temporal exploration of viscous and gravitational fingers. We first present how to detect fingers and branches automatically in space. The viscous and gravitational fingers are ill-defined in the literature. A science paper in 2018 reported that the finger feature is hard to define, but if you see it, you recognize it immediately. Previous works usually used the density thresholding to extract fingers. With a specified density threshold, the regions with density values larger than the threshold are extracted. Although the density thresholding is efficient, it is not sufficient to extract complete branches. For example, here is a finger that contains high-density branches and low-density branches. Also, the high-density branches are connected by low-density regions. If we use a high threshold, although the high-density branches are separated, the low-density branches are missing. If we use a low threshold, the low-density branches are preserved, but the high-density branches cannot be segmented. We propose a ridge-guided finger detection. Ridges originally are structures of surface topography. For example, in this picture, they are the mountain's sharp crusts. Then the mathematical properties of ridges were studied. Ridges of a smooth function are a set of curves or surfaces whose points are local maxima of the function in certain dimensions. In computer vision, ridge detection is used to detect human fingers in 2D images. For example, we can detect the ridge-like curves from the terrain defined by pixel intensities to extract human fingers. Since viscous and gravitational finger structures are local high-density regions in 3D space, we propose a 3D ridge-based detection for the fingers. Rather than detecting ridge lines, we detect ridge fossils, namely the fossils containing ridges. The ridge fossils produce more robust features and capture the branching structures of fingers well. Ridge points are local features that satisfy two ridge criteria. The first ridge criterion constrains the directional derivatives of density function, ensuring the detected points are extreme points. The second criterion requires two eigenvalues of the Hessian matrix defined at detected points, and constrains the two eigenvalues to be smaller than zeros. ensuring the detected extreme points are local maxima in 2 dimensions. Based on the ridge criteria, our ridge Voxel detection consists of 2 steps. First, since the original density function in space is unknown, we approximate the density function. For each Voxel, we establish a second-order Taylor polynomial based on neighboring Voxels' values to approximate the density function near the Voxel. The second-order Taylor polynomials can also provide local approximation of the density function's derivatives, including gradients, Hessian matrices, and the directional derivatives. We decompose the approximated Hessian matrix to estimate the eigenvectors and eigenvalues. In the second step, we compute ridge structures using the approximated derivatives and the ridge criteria. Then we examine whether Voxels contain the ridge structures to extract the ridge Voxels. Here we present the results of the ridge Voxel detection. The first row shows the side view of the raw density volume. The second row shows the detected ridge Voxels. The side view indicates that our method can extract both low density and high density branches and separate different branches. For the top view, ridge Voxels can also capture the high density hexagonal cells in the domain's top layers. We dilate ridge Voxels to recover connections between closed branches. Until achieving the maximal connections of finger branches, we call these finger calls. We continue dilating the finger calls until achieving complete volume, enabling us to analyze whole fingers in space. We skeletonize the finger calls and can get the connections and height of finger branches. The skeletons can show geometric features of fingers and branches clearly. We then present visualization designs to check time-varying fingers and branches. To visualize the characteristics of finger branches, we draw geometric glyphs. For the side view, scientists are interested in the connections between branches. We apply planar visualization to draw branches as vertical lines and draw connections between branches as horizontal lines. For the top view, scientists are interested in the spatial distribution of branches. We draw branches as Voronoi cells, which can clearly show branches' positions and visualize the sizes of branches in space. Next, we embed the glyphs of finger branches into a tracking graph and create the geometric glyph augmented tracking graph to help track finger branches. In this tracking graph, each row contains fingers and branches at a time step. The temporal related fingers are connected through links. Colors represent 3 temporal events: orange for growth, green for splitting, and purple for merging. The thickness of links encodes sizes of overlapping volume between related fingers. Moreover, crossings between links are minimized to reduce the visual clutter. We further designed interactions for tracking branches, which are explained in case studies. We then present the case studies and the interactions by using our developed techniques. We constructed a visual analytics system consisting of 6 panels. The 5 panels in the top row are for the spatial exploration of fingers. The tracking graph in the bottom row is for the temporal exploration. The first panel is used to select a depth of interest. Then the second panel presents the density field slice of the selected depths. The projection panel shows the distribution of fingers in space by representing fingers using convex holes, and support the selection of interest fingers. After scientists select a finger, the finger volume and skeleton are shown in the 4th and 5th panel. The selected finger will also be highlighted in the tracking graph. 2 Earth scientists used our system to perform case studies of fingers. In the first spatial exploration case, scientists were interested in whether fingers tend to form along with boundaries of the top hexagonal cells or form below the centers of cells. The finger skeleton can facilitate scientists to answer this query quickly. By rotating the finger skeleton, scientists can quickly find that this finger grows along with the top cell boundaries rather than forming below the cell centers. The next case is for tracking branches using glyphs in the tracking graph. We highlight a finger with a merging and a splitting event. Scientists can hover over links to identify the temporal correspondence between branches after the merging and the splitting events. Here, this case shows how to track branch volume. A finger with a merging event is labeled. Finger A and finger B merges into finger C at the next time step. By clicking A, scientists identify A had two long branches and a short branch. The volume of C contains the branches from A and B. Scientists hypothesize the left volume of C comes from A. Then by clicking the link between A and C, the volume of C that comes from A was shown for branch volume analysis. In conclusion, we have two contributions. First, we propose a reversal-guided finger detection to facilitate the spatial exploration of fingers and branches. Second, we designed a geometric glyph augmented tracking graph to identify temporal events of fingers and track branch with interactions. Thank you for listening. We also thank our funding providers. I would like to take a few questions.